Hello everyone, I'm Xin Zhao, a faculty member in the Horticultural Sciences Department at the University of Florida. On behalf of the Solanaceous Evaluation and Growing System Working Group for the USDA HCRI Vegetable Grafting Project, I'm presenting a brief report today to highlight the research conducted by our team members. The Solanaceous Evaluation and Growing System Working Group includes researchers from 7 universities and USDA ARS. Building upon findings of rootstock evaluation and grafted vegetable production system development from the Phase 1 project, our work in this Phase 2 project is focused on maximizing the positive impacts of grafting technology and grafted plants on U.S. vegetable industries. Our research addresses 2 main objectives. By assessing different rootstocks, we are documenting in-field responses of grafted plants to major biotic and abiotic stresses. The second objective is to identify the separate and additive or synergistic effects of grafted plants when used in conjunction with soil and crop stewardship practices and tools, including high tunnel production, organic production, reduced tillage, microbial inoculants, and other integrated systems, and to optimize off-farm inputs such as water and fertilizer. In this Phase 2 project, we're also expanding the application of grafting technology to California processing and fresh market tomato production systems. Now, I'm going to discuss specific research work of solanaceous vegetable grafting carried out by our working group members. And we look forward to your feedback as we continue to improve integrated and cost-effective use of grafting technology to serve the vegetable industry. At North Carolina State University, bacterial wilt and verticillium wilt management is a focus of their work on disease-resistant tomato rootstock evaluation and selection. For bacterial wilt, low diversity of the pathogen in North Carolina and the southeast U.S. suggests available rootstocks with resistance should be generally useful in this region. Extensive work has been conducted to demonstrate zones of bacterial wilt resistance, and assays were developed for higher throughput screening that helps capture these complex resistance traits in breeding programs. Greenhouse and field trials have been performed to identify commercial rootstocks for controlling bacterial wilt, although some regional differences were also observed. Resistant breeding lines are under development in North Carolina and Ohio. Some of them were not only resistant to bacterial wilt, but also produced the highest yield. 2 resistant genotypes highlighted here are also highly vigorous, indicating that they may be suitable for tomato grafting as rootstock. Verticillium wilt is the #1 reason growers fumigate in the mountain production region. Rootstocks do not confer resistance to verticillium wilt, but rather a tolerance enhancing yield and fruit size. In grafting experiments, genotypes developed in Japan with resistance to race 1 and race 2 did not perform well in North Carolina, as race 3 was found to be the major strain. Whole genome sequencing was conducted, and novel plant genetics conferring field resistance have been identified in North Carolina breeding programs, and QTLs are also being discovered for future rootstock and hybrid breeding programs. Tomato grafting research in North Carolina also included comparative analysis of rootstock morphology beyond disease resistance. Root morphological differences are present, and these can be quantified and translated into other parameters like rootstock adaptation to stress conditions. Focused on drought stress with the work conducted in growers' high tunnels, a robust rootstock like Beaufort was found to nearly triple water use efficiency at the half irrigation rate. The pronounced yield increase also translated to an increase in revenue, together with a significant reduction in water use. In another 2-year study, grafting high-yielding tomato cultivars onto vigorous rootstocks has been shown to increase marketable yield in the mountain production region on land with no known disease pressure. Pinching plants young and training 2 leaders tended to increase yield, while pruning tended to decrease yield. Grafting work at Washington State University has been focused on verticillium wilt control by screening publicly available eggplant and pepper rootstocks developed by the World Vegetable Center in Taiwan. 14 rootstocks for eggplant grafting and 3 rootstocks for bell pepper grafting were tested in inoculated fields. For eggplant, all rootstocks were susceptible to verticillium wilt, and yield of grafted eggplant did not differ from non-grafted plants. For pepper grafting, none of the treatments displayed symptoms of verticillium wilt, and there was no difference in yield among treatments. Researchers from the USDA ARS in Fort Pierce, Florida, have conducted several years of greenhouse and commercial field trials aimed at developing grafting strategies for managing pathogen complexes in solanaceous vegetable systems. Root knot nematodes are a significant problem for the production of solanaceous vegetables in the Southeast. More than 25 tomato and pepper rootstocks were evaluated for resistance to multiple root knot nematode species and were tested to determine if resistance held up under high temperatures. Grafting onto nematode-resistant rootstocks increased early plant growth and resulted in higher marketable yields in tomato production. Soil nematode populations were lower under resistant rootstocks, and grafted tomato and pepper plants could be combined with fumigants that had low nematode control efficacy and effectively increase production output. In some cases, numbers of other plant parasitic nematodes increased in roots and soil when root knot nematode-resistant rootstocks were grown. Tomato rootstocks were also screened for Fusarium wilt resistance to multiple races, especially race 3. Rootstocks were identified that provided simultaneous control of both Fusarium wilt and root knot nematodes. In current experiments, resistance is being tested under high temperatures. Heirloom tomatoes were grafted onto virus-resistant hybrid tomatoes as rootstocks in order to determine if virus resistance was transferable to the scion. Although resistance was not translocated, grafted plants grew significantly faster and produced higher yields than non-grafted plants. Hypersensitive necrosis was observed in the commercial field when plants were infected with tomato mosaic virus, and the rootstock had high resistance to the virus while the scion was susceptible. This is consistent with results reported by researchers in Japan. Additional work indicated that rootstocks with the TM1 allele for resistance were compatible with all scions, and the hypersensitive necrosis did not occur when the plants were infected with TOMV. Similar work has been conducted with grafted peppers with a focus on resistance to pepper mild monovirus and tobacco mosaic virus. When inoculated with TMV, symptoms in the scions were not affected by the resistance of the rootstock. This research is ongoing. Pepper grafting work also addressed management of Phytophthora blight and soil salinity, 2 threatening problems in Florida pepper production areas, especially in the south. Disease increased under moderate level of salinity in susceptible varieties, but resistant rootstocks maintain healthy plants under moderate and moderately high salinity stress. The Vegetable Production Systems Lab at The Ohio State University has experimented with solanaceous grafting and grafted plants for almost 10 years. In the last 7 to 8 years, it has organized its work around the need to enhance the value of grafted plants as both sources of income for propagators and production tools for fruit producers. The research topic on grafted plant management has involved experiments testing different approaches across the spectrum of soil-based production systems. Specific focus areas have included pruning and training, fertility and irrigation programs, performance in reduced tillage systems, and in combination with microbe-containing crop biostimulants. Regardless of the experiment, plant effects on system variables were documented, such as productivity and efficiency. The preliminary conclusion is that additional improvements in grafted plant management are not only possible, but advisable in order to ensure the continued upward trend in grafted plant use. The working hypothesis is that improving grafted plant management, regardless of whether disease is present, and perhaps especially when it's not, will drive further adoption. The value of grafted plants in situations where the rootstock is resistant or tolerant to a clearly devastating soil-borne disease is well established. However, for many other growers, answers to the questions of whether using grafted plants pays when soil-borne disease is either absent or not severe, and what adjustments in management are needed to maximize returns on investments in grafted plants are less clear. Work at Kansas State University is focused on coordinated rootstock trials to identify vigorous tomato rootstocks for increasing productivity with little to no disease pressure, particularly in high tunnel systems. 10 rootstocks have been evaluated in over 50 on-farm and on-station trials. Grafting has become an effective approach to improving local tomato production and farm profitability. Additional research on scion rootstock compatibility also provided growers with readily applicable information about tomato rootstock performance in maximizing fruit yield. Tomato grafting work in Pennsylvania also has a focus on high tunnel systems to examine the use of vigorous rootstocks for enhancing crop productivity to extend the crop cycle and overcome soil-borne pests and pathogen issues, as well as abiotic stress conditions. Research has been conducted to evaluate the rootstock response to alkalinity stress, which is commonly encountered in high tunnel production systems in Pennsylvania. It was observed that using a vigorous rootstock can help enhance plant growth and increase yield at high alkalinity levels. An ongoing study is looking at the growth, yield, and nitrogen uptake response of grafted tomato plants to increasing levels of nitrogen supply by fertigation in a high tunnel production system. It's aimed to define optimal nitrogen rates for grafted and non-grafted plants under protected environment in Pennsylvania. Research during the Phase 1 project at University of Florida was focused on open field tomato production and demonstrated the great potential of using vigorous interspecific hybrid tomato rootstocks to increase fruit yield and improve nitrogen uptake and use efficiency. Work in this Phase 2 project has been shifted to integrating grafting and high tunnel technologies to identify the role of grafting in promoting organic tomato production. Greater benefits of grafting were found in high tunnel versus open field organic tomato production as a result of increased root knot nematode infestation in high tunnel systems and the overall improvement of fruit marketability in high tunnels. Planting date could also play a role in determining performance of grafted plants in high tunnels due to the environmental influence on crop performance. In the high tunnel grafted grape tomato trial, all 3 rootstocks evaluated increased fruit yield with early planting, while rootstocks did not show a significant impact on fruit yield with late planting. Regardless of planting time, using vigorous interspecific hybrid rootstocks led to an increase in average fruit weight. By conducting greenhouse and high tunnel experiments involving a diverse range of rootstocks, rootstock-scion interactions are being categorized to gain more understanding of the rootstock-scion synergy. In addition, rootstock trials without grafting have been carried out to characterize growth and development patterns of different types of commercial tomato rootstocks To help better classify the rootstock impact on tomato growth and yield performance towards more efficient use of rootstocks. A team of 3 University of California farm advisors conducted the first public field trials of grafted processing tomatoes in California. 6 trials were conducted in cooperation with 4 different growers within commercial processing tomato production fields. The team initially focused on rootstock assessment, while reduced plant population treatments were added beginning in 2019. Major soil-borne diseases in the region are Fusarium wilt risk 3 and verticillium wilt, although these have not been a focus for most of the field trials. Grafting was conducted in collaboration with 2 California transplant nurseries. In 5 trials evaluating 11 rootstocks, grafting increased yield by an average of 16%. Maxifort, which was included in all 5 trials, increased yield by 23%. There was a wide range in performance of various scion rootstock combinations, with yield increases varying from 0 to 55% over the non-grafted scion cultivar. While high-vigor rootstocks can increase yield, there are concerns about the large vine size and associated delays in maturity. 2019 results on in-row spacing suggest that grafted plant densities half the normal density are sufficient, but the adoption of grafting remains cost-prohibitive at current prices. Given the low commodity price for processing tomatoes, grafting cost needs to be substantially reduced by lowering rootstock seed cost and increasing the efficiency of the grafting process. To date, 3 years of field studies have been completed evaluating rootstocks for heirloom tomatoes in open field production in California. 2 heirloom scions and 6 rootstocks were evaluated. Grafting was done by plat connection in Southern California. Heirloom tomato production varied from year to year. None of the trials were in fields known to have soil-borne diseases. Across the 4 trials conducted in 3 years, the ranking of each rootstock also varied widely. A consistent trend was hard to elucidate. This work is still ongoing, and data from all trials will be analyzed together to help gain a comprehensive understanding of rootstock effects on fruit yield components and production curves. That concludes the brief report from our Solanaceous Evaluation and Growing System Working Group report. Please feel free to reach out to individual researchers regarding specific studies. Again, we would like to thank the USDA SCRI program for supporting this project. We appreciate all the great support and help from our partners, and thank you to our Stakeholder Advisory Board. We look forward to your feedback. Thank you.